Hello students, welcome to F.Y.B.Sc class. Today we are going to discuss about the topic bacterial growth curve from the chapter Microbial growth of F.Y.B.Sc Microbiology paper 2 MB122 Microbial cultivation and growth. The learning objectives of this lecture is to understand the growth curve of bacteria. How you can define a growth? If you find any increase in the cellular constituent or if there is increase in the number of population, then that is called as the growth. So growth can be defined as increase in cellular constituent, and because of the increase in cellular constituent, it may increase in the number of organisms as well as increase in the number of population, then that one is known as the growth. Now, the growth is not related only with the increase in the mass, but because the cells can increase their mass. So by increasing the storage product like the glycogen or the lipid, and therefore if these storage products are increased, then it leads to the increase in the size as well as weight of the cell. Now growth is normally followed by a cell division, where in case of the bacterial cell, one cell will divide to give rise to two number of bacterial cells, and therefore when in case of the bacterial cell, growth is followed by increase in cell number. Now, as you have already know that bacteria, they divide by the process of binary fission, where a single bacterial cell, it may give rise to two cells after developing a transverse cell wall. Now, a bacterium can completely produce new organism or complete duplicate of its cells, and that period is a very short period. That will also depend on the bacteria, and the shortest time which was reported is 20 minutes. Now once there will be increase in the bacterial cell, after that you will find that at the end of that phase there will be decline in the viable or the living cells. Now, so between each of these phases, there is a transitional period. The growth curve is divided into different phases. The first most important division is the log phase, logarithmic growth phase, stationary phase, and the logarithmic death phase. Now these four phases are again divided into different phases. Just like lag phase will be divided into two phases, initial stationary phase and the phase of accelerated growth. Logarithmic phase will be divided into two phases, logarithmic growth phase, phase of decreasing growth rate. Stationary phase is also known as the maximum stationary phase. Logarithmic death phase can be divided into three subphases: phase of increasing death rate, logarithmic death phase, and the survival phase, which is also known as the phase of decreasing death rate. So we'll discuss each stages of this growth curve in detail. So to study the growth of the population, we have to study the growth curve of a microbial culture. Now for that we have to inoculate or cultivate this microorganism in a liquid medium. Now for that mainly batch culture or closed system will be preferred. Now closed system or the batch culture is the culture where we have added a single batch of nutrient medium, and therefore that type of the culture is known as the batch culture or the closed system. Now, in this case of the batch culture, as medium is added only at one time, and therefore nutrient concentration will be declined when the organism they start to utilize it. And in addition to decline or the decrease in the concentration of nutrient, concentration of waste product will also increases. So we can plot the growth of microorganism as the logarithmic of number of viable cell versus incubation time. So the growth curve of a particular bacterial cell have four distinct phases. So here is the growth of population, number of cells versus time. So it mainly divided into the lag phase, as you can see. After the lag phase, the organism will enter the next phase, that one is also known as the log phase, logarithmic phase or the exponential phase. After this, organism will enter the third phase, that one is known as the stationary phase, and there will be decline in the number of cell. That phase is known as the death phase or the decline phase. So this figure is indicate the microbial growth curve in closed system. First, we'll discuss about the lag phase. What kind of the changes will take place within that cell? So, when any organism is introduced into culture medium, then organism will not start dividing. So, they require some time, and after that period only, you will find increase in the number of cells, and that period is known as the lag phase. So when organisms are introduced into the fresh culture medium, no immediate increase in the cell number occurs, as organisms require some time to adapt to that environmental condition, and this period where no growth is found, that is known as the lag phase. So in this case, in the case of the lag phase, you will not find any kind of the cell division, and there is no increase in the mass, and because the cell is busy in synthesizing new components. So lag phase is the most important phase. Now the cells they are getting old, or because of the lack of energy which was provided in the form of the ATP, essential cofactor and the ribosomes. So they need to synthesize all these components to enter the cell division, and therefore the lag phase is play very important role, because during this phase, although there is no increase in the cell numbers, but cells start to synthesize all the essential molecules which are required for the cell division. In addition to that, the nutrient medium which are using, they may also differ from the organism. We may previously grow the organism in the different medium, and now we are using the another medium, so that to adopt they have to synthesize the enzyme. By using that, they can use variety of nutrients which are present within that nutrient medium. Without enzyme, they cannot utilize the nutrients which are present within new medium. So the organisms they have been injured, and they may also require the time to recover. Now the cells retool, they will replicate their DNA, begin to increase in the mass, and finally divide. So the phase lag phase, it may vary in length. That will depend on the condition of organism, as well as that is also depend on the nature of the medium. As compared to other phases of the growth curve, the lag phase, it is quite long. If the inoculum or the organisms which you have added, they are from an old culture, or if you make the use of refrigerated organism, then they also requires more time to adjust to the new environmental or new cultural conditions. Now, inoculation of culture into a different chemically medium, it also results in the longer lag phase. So, if you have transferred young culture to the fresh medium of the same composition, then in that case the lag phase will be short or the absent, because organisms, they are utilizing the same nutrients as we are making the use of same nutrient media. So in that case there is no need to synthesize the enzymes which are required to utilize the nutrients which are present within that culture medium. So in this case, the time of the lag phase will be short. Next phase of the growth curve is the exponential phase. Exponential phase is also known as the log phase or the logarithmic phase. Now, when the organism enters the lag phase, they synthesize all the molecules which are required to utilize the nutrients which are present within the new fresh medium they have added, as well as to carry out the cell division. Whatever components they will require, all were synthesized during the lag phase. In the case of the exponential phase, organisms now have synthesized the enzymes. By using that, they can utilize the nutrients which are present within that medium. So the organisms are growing and dividing at the maximal rate, and the growing rate of the organism will depend on their genetic material, as well as the nature of the medium in which we have added, and the condition under which they are growing. Now, in case of the exponential phase, the rate of growth is constant. That is, organisms, they are dividing and doubling in numbers. But in this case, if you look at the growth curve of individual organism, then it will be differ slightly. But in case of the exponential phase, the growth curve rises smoothly. So in case of the, if we talk in terms of the chemical and the physiological properties of the organism, then the population is present in the uniform state. That is, they have all the synthesized the enzymes. Now they are ready to grow and divide, and therefore you will find the increase in the number of organisms. So exponential growth is also known as the balance growth, because all the cellular constituent, they are manufactured at the constant rate, and therefore exponential growth is also known as the balanced growth. Now, if there is a change in the nutrient level, or if there is a change in the environmental condition in which they are going, then unbalanced growth can also occur during the exponential phase. So exponential growth is related with the increase in the synthesis of all the cell component, and therefore a new balanced state is reached. Now during the exponential growth, bacteria are transferred from the nutritionally poor medium to the richer medium. Then in that case you can find the change in the response curve. Now, in this phase, the cells, they will construct new ribosomes, so can they increase their capacity to carry out the protein synthesis process. So once the proteins are synthesized, if there is an increase in the protein as well as then they'll start the synthesis of DNA, and you will find reproductive rate will be increased. Now in this case, unbalanced growth will be found in the exponential growth curve if the bacterial population, if you have transferred it to the poor medium, that is which lack the essential nutrients which are required for the growth of that organism to be take place. So if the organisms they have been added to the medium which they have been previously encountered, then the log phase will be reaches it at highest peak and the lag phase will be shorter. If we have transferred the organism to the poor nutritional medium or the nutritionally inadequate medium which lack the essential nutrient, then organisms will try to synthesize the enzymes by which they can utilize the nutrients which are present within that medium, and therefore the cell division as well as DNA replication rate will be slowed down, but net protein and RNA synthesis process will also slow down in this case. Next phase is the stationary phase. Now in the lag phase, organism try to adapt to the new cultural as well as environmental condition and try to synthesize the component which are required to utilize the nutrients as well as to carry the cell division. During the log phase, which is also known as the logarithmic phase or the exponential phase, organisms synthesize all the components, so they utilize the nutrient in a faster way. So you will see their reproduction rate is also increased, and in which results in the increase in the number of cells. After that, the organism will enter the next phase of the growth curve. That one is known as the stationary phase. Now, in this case, although population growth will be ceases and growth curve will become horizontal. So, this stationary phase will result in the population level of around 10 raised to 9 cells per ml. So other organisms they do not reach such a high population density like the protozoan and the algal culture. If you have studied a growth curve by using these two culture, then the maximum concentration of the cell will be around 10 raised to 6 cells per ml. So the final population size in the stationary phase will depend on the availability of the nutrients and different factors, as well as it also depends on the type of organism which you are going to culture. In the case of the stationary phase, the total number of viable or the living organisms remains constant. So in case of the stationary phase, you will observe there will be a balance between the division and the cell death. But during this phase, the organisms they remain metabolically active. Why this microbial population they enter the stationary phase? The first most important reason is the limitation of nutrient. During the lag phase, they synthesize the enzyme by which they can utilize the nutrients which are present within that culture media. When this organism they will enter the log phase, they start to utilize the nutrients at the higher rate, and that results in the decrease in the concentration of nutrient within that medium. So when they enter the stationary phase, there will be decrease in the concentration of essential nutrient, and therefore population growth will be slowed down. If your organism is aerobic, then it may require the oxygen. So in case of the organic aerobic organism, their growth can also be limited by the availability of the oxygen. Oxygen is not very soluble and therefore it may weakly depleted or its concentration may be slowed down. Its concentration will be higher only at the surface of the culture, and therefore the cells which are present at the bottom of that culture medium, they are unable to grow unless the culture is shaken or aerated. So this graph will indicate the effect of change in limiting concentration on the microbial yield. So during this stationary phase, the organisms are metabolically active. So we can take the example of streptococci. So this streptococci, when it enter the stationary phase, they will start to produce the organic acid like the lactic acid and other organic acid from the sugar which was present within that medium. And because of the secretion of the acid, the medium will become acidic in nature, and because of that also growth is inhibited. So if the depletion of the sugar supply, that is if the concentration of sugar is decreases, then also these streptococcal cultures, they may enter the stationary phase. So the bacteria, they enter the stationary phase in response to the starvation, that is the depletion or decrease in the concentration of essential nutrients. So this oftenly occurs within the nature because many environment, they have very low nutrients levels, and therefore when the nutrient concentration will be slowed down at that time, it will affect the growth rate of the organism. Next phase is the death phase. So organisms have utilized all the nutrients which are present within that medium. In addition to that, the toxic waste product will also, their level will also increases within that medium. So because of the cumulative effect of deprivation of nutrients and the buildup of toxic waste product, it leads to the decrease in the number of viable or the living cells, and this is the most important characteristic of the death phase. So death of the microbial population will occur at the exponential phase. So in the case of the death phase, the viable cell count will be whole because the cell number will remain constant as the cells, although they will die, but they are not. So to decide whether the bacterial cell is viable, they, so to decide whether the bacterial cells are viable or living is to inoculate them into the fresh medium. If they do not grow and reproduce, then we can say the bacterial culture is dead. So, death can be defined as irreversible loss of the ability to reproduce. So most of the microbial population, they die in a logarithmic fashion. So death rate may decrease after the population has been drastically reduced. And this is because of the survival of resistant cell, those who are able to withstand the low nutrient concentration. The death phase curve may be complex. These are the references You can refer a textbook on microbial cultivation and the growth published by the Success Publication, and the authors are Dr. Sunita H. Patil, Dr. Sucheta N. Patil, and the Prajakta Pachorkar. Thank you.